Hi everyone, it's Mr. Sinti, and I have the honor to discuss with you today the idea of how biological molecules can act as catalysts. That's right, enzymes. And so many chemical reactions in biology require the regulation of enzymes. And so an enzyme is a biological molecule that could either speed up or slow down a chemical reaction. And you're like, so you have a couple of questions about this. You're like, what biological molecules are we talking about? Why would we need to speed up chemical reactions or slow them down? Well, all of this will be part of our discussion. And so, one of the things that I want to say is that an enzyme acts as a catalyst. Now, a catalyst doesn't always have to be an enzyme. Like you could be a catalyst, for example, if you're trying to introduce two of your friends to get together for a relationship. You would be sort of like a cupid. You would be getting them together, speeding them up, introducing them to one another. So you'd be acting as a catalyst. A match can be considered to be a catalyst by burning wood, for example, getting it going. So it's some kind of agent, whether it be chemical or person. It's an agent that changes the rate of a reaction. Either it speeds it up or slows it down. But it doesn't get consumed in the chemical reaction itself. That's kind of interesting. It can be therefore used over and over again. And so an enzyme is often considered by most students of biology as a protein, and sure, most of them are. But RNA, ribonucleic acid, can also be a catalyst. And it's like, well, what do those two things have in common? They both have the ability to have tremendous three-dimensional shapes or conformations that allow themselves to act as a catalyst. You need a complex three-dimensional shape in order to speed up a chemical reaction, and we're going to see that in the video today. And so that's kind of cool. Enzymes regulate the movement of molecules because basically that's what we're talking about in pathways is the movement of reactants to products, and then the products become the reactants of the next pathway. And so an example of this would be our good friend table sugar, disaccharide sucrose. You can see here it's made up of glucose and fructose. If we wanted to digest it, if we wanted to hydrolyze it, we would simply add water. These are the two reactants, here are the substrates. So water comes in and it cleaves that glycosidic linkage, and the product is, of course, glucose and fructose, and there's the water replacing that. And so this is a hydrolysis. Now, this is a spontaneous reaction, meaning that it has a negative delta H and it has a positive delta S. And so it's a release of Gibbs free energy, negative delta G. So all that being said, though it's spontaneous and it's likely to occur, it still requires a little bit of help. And so an enzyme can provide the catalyst. So in other words, this right here is an enzyme, and I don't know if you have any background in enzymes, but we like to call the name of an enzyme, so naming an enzyme, we like to say it's the name of the substrate, so sucrose, and we remove the ose at the end and add the suffix ase. And so when you put this together, it's sucrase, like that. That's the name of this enzyme. And so what's happening here three-dimensionally, you might notice in this cool photo right here, you can see alpha helix, beta pleated sheet, alpha helix, beta pleated sheet. So this is the tertiary or 3D structure of the protein, and it has indentations or alcoves right in here. And so the substrates could fit inside here, and then the products emerge. And so the point is, more of this to come later, but the point is to get the substrates to collide at a lower temperature than they would otherwise without a protein speeding this up. So it provides a small crevice for chemical reactions to occur. And so chemical reactions between molecules are all about breaking bonds and forming new ones. And so the hydrolysis of sucrose is an example of that, and the enzyme sucrose can speed it up. Now, where is that happening in our body? Well, it's happening in our small intestine. Like, for example, food is coming down, principally sugar. And so here's the sucrose right here. This is that disaccharide that we were just talking about. So there it is. And so what's the point? Why break it down? Well, it needs to be smaller into monosaccharides for the cells to absorb them in the digestion. This happens to be simple columnar epithelium, if those of you who know that, you can tell by the free space. And so hydrolysis is happening out here extracellular. And so where's the water coming from? We're either drinking it or drinking something that has water in it, or our own cells can, how about this, release mucus into this area. These are goblet cells because they sort of appear as goblets right here, like little chalices containing mucus right there. And so they're releasing water into this free space, and that's speeding up the digestion. So here's something. How did the enzyme get out there? How did the enzyme sucrase get into this space in the first place? Well, the cells actually produce these proteins, and the proteins get exported to the outside. And so that, I won't get into this right now, though I'm tempted to do so, is that's an interesting dilemma, because a protein, if you're following this conversation, a protein's pretty huge. It's very, 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 very large. And I was just saying that sucrose is too big to come into the cell, but yet somehow the cell is able to secrete something really large as a protein enzyme. And so I'm just going to let that hang and hope that tantalizes you to watch future videos on the export of macromolecules. It has to do with the endomembrane system. That's pretty cool stuff. So in general, enzymes speed up metabolic reactions. So these are all the totality of all chemical reactions in the cell. How do they do it? Well, they do it by, the definition is they do it by lowering the energy barrier. You're like, okay, what's that? Well, if a chemical reaction is going to occur, do you see these reactants down here? Like if this one obviously releases energy right here, so this is an exergonic reaction, so it's a negative delta G. It's going to occur spontaneous. It's going to have a positive delta S. But do you see this right here? The energy of activation, though a reaction is spontaneous, sometimes outside energy is needed, and it prevents a barrier. This energy is needed to break these or stress these chemical bonds. And so what an enzyme does is that it lowers the need for excessive energy of activation. Therefore, cells don't require a tremendous amount of energy to get the reactants to collide or stress them out or break the bonds. So, in other words, you don't need tremendous heat. And so it lowers. So if I were to animate this, I could say, well, an enzyme, if I were to add an enzyme, it's not going to change the Gibbs free energy, but it's just going to lower the amount of activation energy needed. So therefore it lowers the energy barrier. That's what an enzyme does technically. So how does it do that? Well, it makes the reactants a little bit more unstable, and it also causes them to collide more readily in such a small space. As I was saying before, you get these reactants into this small area right here, they're more likely to meet up with one another. So the enzyme sort of acts as a catalyst that way. And so the energy of activation, remember, is that energy needed to get the reaction to go. And so enzymes lower the need for that, but they don't they don't affect the difference of free energy. That's important. And so just to make sure that we're all good with the activation energy, if I can make an analogy to like a car, for example, gasoline, in order to get gasoline to ignite with oxygen, you need a spark plug to do that. And so there's spark plugs in a car. And so that provides the activation energy to release the energy in the hydrocarbon gasoline. Now, as it turns out, it's an exergonic reaction. In other words, it's going to release heat. The products are more random, so it's a positive delta S, and therefore it's a negative delta G, free energy. But yet, it doesn't happen at room temperature because the activation barrier is too high, so you need a spark plug to get it going. So an enzyme basically takes the place of the need of a spark plug, because the thing is, though heat boiling something, burning something are tremendous catalysts for chemical reactions. It turns out that cells don't like a tremendous amount of heat. It's very damaging. And also, proteins can get agitated at high temperature, and therefore they lose their characteristic shape, and so therefore they don't function efficiently, mostly at high temperature. So heat is not a good biological catalyst. And so enzymes lower activation energy. And they do that because the reactants can reach this so-called transition state even at room temperature, or in our case of our body at warm temperature, which is 37 degrees Celsius. So enzymes again don't change the energy display between reactants and products, or the dynamics of energy, but rather they lower the energy of activation. And so that's pretty important. So enzymes are, another characteristics of enzymes other than the activation energy is that they're very, very specific. And their specificity has to do with their three-dimensional shape. And I mentioned that this example before of sucrase as being the enzyme that speeds up the hydrolysis of sucrose. There's many, many thousands of different enzymes that are needed by a cell because each substrate binds to a particular enzyme. That's incredibly important because these enzymes are kind of like traffic lights on all of the roads in a big city. And so you don't want the enzyme capable of speeding up different kinds of chemical reactions. You want specific reactions to be sped up or slowed down because the traffic flow is going to be different from time to time in different areas. And so substrates bind to an enzyme very, very specifically. And so it's only sucrase that has eyes for sucrose. And so you can see here in this computer-enhanced model of an enzyme, all of these sort of blueberries are amino acids, and here's the substrate. Only this one substrate is capable of fitting into this one crevice of the protein. This little area right in here where substrates fit in, that's very, very specific, is referred to as the active site. That's a very important term, the active site. It's where it's going down, the chemical reaction. Now, what's interesting about it is there's a temporary bonding that occurs, and I'm going to talk about that a little bit later, that occurs. And so the active site is typically like a pocket or a groove in the protein three-dimensionally, or an alcove for which the substrates fit. And the specificity of the enzyme has to do with that active site. And so that's very, very critical that occurs. And so when the substrate does bind to the active site, it leads to a interaction. Like I'll see if I can draw this. It leads to an interaction between the substrate and the protein such that it causes the protein to change itself and therefore stress the substrate. And so therefore this is referred to as an induced fit. So the enzyme changes the shape, leading to a tighter fit, and therefore it brings about the chemical reaction more readily. It allows the two reactants to collide more easily, or it allows for bonds to be broken more easily because of that temporary docking with the active site. And so it's kind of like, I'm prone to go a little overboard, as most of you know from watching previous videos on analogy, but I like analogy. The enzyme is like a baseball glove, and the pocket is like the active site, because this is where the substrate or the ball fits in, and this is like the point of a glove is to catch the ball. That being said, you can't disregard the other structures in a baseball glove because those facilitate the active site. You're like, well, this is the most important part, the pocket. Well, where you put your hand in is also pretty critical too, because that's enabling you to, when the ball cuts into the pocket, enable you to squeeze the glove and to capture it or catch it. And so all of that's kind of important. Most people think of an analogy as being a lock and a key. In other words, the the lock is very specific, and the key is the substrate fitting into the lock as well. So that's a good one. So in most cases, the substrate right here in this case, we're still going with the sucrose. Here's glucose and here's fructose over here. And so what's happening is, how does it bind with the protein? Well, it's usually held temporarily by hydrogen bonds and ionic bonds between the R groups that are protruding in the amino acids found in the three-dimensional active site. Now remember, this is like a glove. It goes all the way around, and this is like the ball. And so the R groups in the amino acid in the protein are able to interact with the substrate and therefore stressing it. And so here's what I was talking about in terms of, like, this would be sucrose. This would be sucrase, the enzyme, and this would be sucrose. And what's interesting is, you're like, well, doesn't any key fit into any lock? No, the shape, this is what we're talking about. The shape of the molecule is particular to the shape of the enzyme active site. And so it's a good analogy, lock and key. Here's another picture of this. Here's the enzyme or the glove or the lock. Here's the substrate sucrose. So here it comes. It goes in like this, and this is happening, remember, outside the cell. Where did this big protein come from? It came from our intestine. It's secreting this. Why? To digest our sugar. So water comes in, it clips or breaks the bond, and then the products come out, glucose and fructose, those two monosaccharides. Notice something very important here. The enzyme itself didn't participate per se. In other words, it wasn't consumed. It participated in a sense that it stressed the bonds and reached the transition state in the induced fit, but it didn't get altered itself, and so it can continue to be reused. So just a little bit of sucrase can digest a tremendous amount of sucrose. And so how did it do it? It reduced the activation energy necessary to catalyze that chemical reaction. And so therefore, this reaction can occur at a lower temperature. Really important. And so it turns out, as I was saying, that it's just a single enzyme can catalyze thousands of reactions in a second. It's really fast. And so this helps to speed up all metabolic reactions if necessary. And so enzymes are reusable because they don't participate. And so the enzymes are really cool. They could go either in the forward direction to the products, or the products can go back to the reactants depending on the equilibrium of the chemical reaction, if you're familiar with that concept from chemistry. If there's a lot of substrate, it's going to favor the forward reaction. If there's a lot of product, it's going to favor the reverse direction, the Le Chatelier's principle. And so here's another cool example of a chemical reaction. We could say this is a classic. This is the very first chemical reaction in the metabolism of glucose, or cell respiration, or at least carbohydrate metabolism. So let me see if I can go with this. So here are the substrates, glucose. So first thing that happens is when, let me draw the cell membrane like this, and here's a protein. So first thing that happens when glucose travels across a protein into the cell, here's glucose, right there, there's the substrate. It's greeted, here's the chemical reaction, it's greeted by adenosine triphosphate. So there it is, ATP. There's adenine, the ribose sugar, and a triphosphate. And so the way we draw that is it's glucose plus ATP. And what's happening in the chemical reaction is this enzyme right here, I'll show it in red right here. I'll just put it above here like this. This enzyme called hexo, because glucose is a 6-carbon sugar. So hexokinase. A kinase is an enzyme capable of movement, transferring a phosphate from ATP to glucose. So there's the chemical reaction. So the product of this is glucose phosphate. And so there it is, glucose 6-phosphate, in reference to where it is on the what carbon the phosphate is attached to. And so what else is a product in this chemical reaction? Adenosine diphosphate, because it's losing one of its phosphates. So here are the products, here are the reactants, here are the products. And how this occurs is the enzyme hexokinase speeds up that chemical reaction. It's pretty cool. And so, again, just to finish with this notion, how is it that they work? After watching this, can we still answer that? Can we answer this question? In other words, here's a protein. Here's this red active site. This is pretty cool. You can see the 3-dimension. Maybe this yellow dot and this green structure are the substrates. How does it actually work? Well, enzymes use a variety of means to lower the activation energy. Okay? So, in other words, the speed necessary for the reactants to collide, so less heat is needed. And so the way in which we think the active site works is that the substrates fit in just the right, and I'm going to circle these, just in the right orientation for the reaction to occur. In other words, it holds them in just the right 3-dimensional shape so that the collision can occur more readily. So the correct orientation is one way in which we think it works. We also think that the interaction between the R groups in the active site, in other words, hydrogen bonds or ionic bonds, interacting with the substrates puts a stress on the bonds and makes them easier to be broken and reach that transition state. So this induced fit is what we're kind of talking about right here as well. So orientation and induced fit. And then, frankly, it could just be the environment itself. It's a conducive micro environment. In other words, they're more readily to collide since they're in a small space. And also, perhaps just the nature of those amino acids and the substrate can create sort of a negative environment or a slightly acidic environment, which will then cause the chemical reaction to occur more readily. So that's pretty cool. Enzymes are some of the most important biological molecules of all. And so I really hope that you enjoyed it and understood a little bit about how enzymes are biological catalysts, both proteins and RNA. Because let's not forget about RNA again, because the large ribosomal subunit acts as a ribozyme. In other words, RNA as an enzyme catalyst that speeds up the formation of polypeptides. That's pretty important too. So hope you enjoyed this podcast on enzymes. Thanks for watching.